Okay, welcome back. Hope you all had a nice lunch break. I'm really happy to introduce Deanna Barch. Deanna, we had hoped would be at the symposium that was actually canceled because of COVID. She was originally scheduled for that meeting, and so it's great to finally welcome her to this platform. I've known Deanna for a really long time. I've known her since right after graduate school. And Deanna really has had three careers. The first career was as a cognitive neuroscientist. And I remember hearing about her from my friend Jonathan Cohen, who she did a postdoc with when Jonathan was at Pittsburgh and Carnegie Mellon, and said this amazing clinical psychologist who is doing cognitive neuroscience. And those were the days, pardon my age, when it was very rare. In fact, Deanna may have been really the first to do that in that kind of really rigorous way. And after her postdoc, she went on to WashU and has been there ever since. And it's really been a thrill for me from afar to just see her career flourish in so many remarkable ways. Deanna's work is focused on the understanding of the interplay among cognition, emotion, and the brain. In psychopathology, she devoted a large chunk of her career to studying schizophrenia. More recently, she has been studying depression and related disorders, and she uses many different methods, including behavioral methods that are informed by her expertise in cognitive neuroscience. She uses clinical methods, and of course, she uses an extensive array of different imaging methods in that work. And most recently, she has also been focusing a lot on the impact of early adversity and charting how early adversity really gets under the skin, both poverty, stress, disparities in access to healthcare, all of which impact our mind and our body and our brain in all kinds of ways. And one of the other things that she's doing a lot recently, which I really applaud tremendously, is she's studying interventions. And she's studying how they can affect the specific mechanisms that she has characterized as going awry. She's the chair of the Department of Psychological and Brain Sciences at WashU. She is the Gregory Couch Professor of Psychiatry. And in addition to appointments in psychology and psychiatry, she also has an appointment in radiology at WashU. And so she comes with lots of wonderful things in her toolbox, and I'm looking forward to hearing her talk. And let's all please welcome Deanna.
All right, can you hear me okay? Great. Thank you for that lovely introduction. I have many memories of at the time meeting Dr. Davidson at conferences when I was a graduate student. So it feels very full circle to come here and talk. And I've never been to the Wisconsin Symposium on Emotion, even though I've heard wonderful things about it for so many years. So I've really enjoyed the day so far, and I look forward to the rest of the day, the posters, and tomorrow. And I'm going to talk as Richie noted, about a somewhat more recent line of research for me, although it's a little bit full circle because most people who knew me earlier in my career definitely think of me as someone who studies adults with schizophrenia. But most people don't know that my very first project in graduate school, my master's thesis, was in kids looking at anhedonia and depression, and it didn't work. And I didn't publish it. And so for anybody who's had that issue, let me tell you, it doesn't stop your career. And I kind of went on to study schizophrenia. And then once I had tenure, I was like, you know, I can go back to studying development. And I'm going to talk about a line of research that I've been doing with just a fabulous set of colleagues trying to understand the sort of psychological and neural mechanisms that might contribute to risks for depression that emerges either early in life or kind of continues on. I'm well trained to disclose my interests. I've been really fortunate to be funded by NIH, NIMH, and NIDA, and NIGMS, and the NIH Blueprint. And I want to start at the beginning, really acknowledging this fabulous team of people, because I'm going to talk about a bunch of different studies with different methodologies that are really challenging. And I don't want to in any way, shape, or form take all the credit for it. So in particular, I want to thank Joan Luby, who is my partner in all things good. She is a child psychiatrist. Mike Gaffrey, who was our postdoc, who's now at Duke. Brent Rappaport, who was a wonderful graduate student, who's now a postdoc with Stu Shankman at Northwestern. Diana Whalen and Kirsten Gilbert, who were postdocs on our T32 and are now faculty in psychiatry, and Catherine Luking and David Pagliaccio, who were my graduate students in the neuroscience program. And Catherine is starting a faculty position as an assistant professor in the fall at St. Louis University, and David is now an assistant professor at Columbia University. So really want to acknowledge them. And as we go along and talk about the different studies, their faces will pop up to just really highlight their critical role in these different projects. And sort of the starting point for this work, to some extent, was saying, well, historically, there's been a lot of work on the behavioral and brain systems that we know are involved in adult and adolescent depression. But there's been much less work historically on depression emerging in childhood or even preschool. But nonetheless, the work in older populations is informative for us thinking about kind of starting points of where to kind of look in kids. And that work has really converged on disruptions in a variety of neural and psychological systems. Let me go here. If you look at work on the neural systems of emotion, right, we've heard a lot this morning about the amygdala. There's certainly a critical role for the amygdala. We've heard about the hippocampus, critical role for the hippocampus and other brainstem nuclei, and their interactions with systems. So these might be characterized as systems that are involved in responding to events that might be salient or emotionally evocative or identifying emotions. And then we have systems such as the orbital frontal cortex, the ventral medial prefrontal cortex, subgenual cingulate cortex, that people have argued might be involved in attentional control of emotion processing. And then we have regions in the brain that are more broadly involved in cognitive control, emotion regulation, and might play important roles in sort of top-down regulation of responses in systems that kind of are more involved in emotion processing or emotion identification. And what we know, oops, I get ahead of myself here. What we know now from a large body of research, and it's been kind of organized in the RDocs framework, or the research diagnostic criteria, is to think about these things in terms of negative valence systems. So systems that are involved in processing and responding to negatively valenced information, sadness, loneliness, systems involved in emotion regulation, or the sort of top-down control over emotion. Also, the connectivity between these regions and circuits, right? That they have to communicate with each other and information has to flow between these circuits in order for people to effectively regulate their emotions. And then we also have positive valence systems, right? Systems that are involved in responding to reward and pleasure, to helping us to have a hedonic experiences in our lives, presumably might be involved in well-meaning and positive affect. And really in the adult literature, there's evidence for disruptions in all of these systems associated with depression, right? Not one single system seems to be involved. And so we started to ask the question about whether disruptions in these same systems were also present in children who either had very early onset depression or who were at risk for depression by virtue, for example, of having a family history of depression, or who developed depression in adolescence. And what I'm going to do is kind of walk through these 4 areas of research in adults and talk about what we've been learning about in kids, about when these things might emerge, what they might look like in children. Spoiler alert, I don't know if it's a spoiler alert, but I was always taught in graduate school that you should tell them what you're going to tell them, and then tell them, and then tell them what you told them. So I'm going to tell you that there is evidence for disruptions in all of these same systems in children at risk for depression, children who've already experienced depression in preschool. So the causes of these things are arising very early in life. And we see evidence as early as preschool of these kinds of disruptions. And I think that, as Richie alluded to, has critically important implications for thinking about prevention and intervention. And I say that because, childhood and development is this critical period of time where you're engaging in both kind of experience expectant and experience dependent learning. And sometimes you can't go back to those periods of time, right? We know there are sensitive periods and critical periods for some psychological and emotional processes. And if kids are going through those periods with disruptions in these systems, right, they may not be learning and having the experiences that they need to develop into healthy adulthood. So the earlier we can think about helping kids sort of shift back on a more typical trajectory, the more they may be able to benefit from these positive experiences. So that's my forewarning. All right, so let me start by talking about whether kids with depression or at risk for depression show disruptions in kind of the negative valence systems, right? Either psychological or neural responses to stimuli that might elicit negative experiences or emotions. And we've studied this in a couple different ways. One of the studies that we've been able to have access to for a number of the studies I'm going to talk about is a now 17-year-long prospective longitudinal study started by my colleague, Joan Luby. So before I even knew Joan Luby, she recruited a sample of over 300 preschoolers that were enriched for early signs and symptoms of depression. There were also healthy children, kids with anxiety, kids with disruptive disorders between the ages of 3 and 6. And we have been working with those families and those kids for the last 17 years. And we just finished the 5th wave of neuroimaging with these kids and their families. And we actually just submitted a grant to follow them up into, into adulthood. So it's been a really rich data set and a wonderful experience to see these kids grow over time. We've done semi-structured diagnostic interviews. We've measured everything under the sun. We have observational assessments of parent-child interactions. We now have, Fitbit data, actigraphy, got everything under the sun. And we started imaging in waves 4 and 5 when the kids are about 8 to 10 years old, and then have done 5 waves over time. I won't talk about all of those waves today, but I'm happy to talk about them for anybody who's interested in some of the things that we've done with that. And way back when, which feels a long time ago now, when we started the first wave of imaging, we looked at the literature, and the literature said, perhaps a very sensitive way to look at responses to negative information is to use a simple task and to not ask people to focus on the emotion. So the literature at the time suggested that you saw the most evidence of differences amongst adults with depression when you didn't orient them to emotion processing. So we used what were called the NIM-STIM at the time, emotionally evocative faces with different emotional expressions, but all we asked the kids to do was to tell us whether they were males or females, right? And in retrospect, I apologize for the gender binary, but that's what we did at the time. So we had kids get in the scanner. We showed them a bunch of faces. We simply asked them to tell us whether they were male or female. And then we looked at brain responses as a function of the emotional content of the faces. And we related that to the children's experience of depression. And what we found in this early study was that the depression severity of kids at the preschool period, so this was 3 to 6 years old, was associated with greater activation in a variety of regions known to be sensitive to responding to negative information. And it was in particular sad faces that seemed to elicit greater activation amongst these children. So when we looked here, we saw greater activation as a function of the severity of childhood depression in the orbital frontal cortex. We saw this relationship remained even when you covaried for comorbid anxiety, externalizing symptoms, even depression at the time of the scan. So it seemed to be particularly related to how severe their depression was at the preschool period, because we were scanning them when they were actually at school age. We also saw it in the striatum. We saw it in the amygdala. So early evidence that kids who had depression in preschool were already showing evidence of sort of increased responsivity of these negative valence systems. Now, these kids had all essentially had somewhat of a diagnosis of early onset depression, but we also know that kids who have a family history of depression are at a greatly increased risk for developing depression themselves, likely for many different reasons. And so we were also interested in understanding whether some of these differences in brain function or behavior emerge prior to the clinical diagnosis of depression in kids who are at risk. So my intrepid graduate students, Catherine and David, decided that for their dissertation, they were going to recruit a whole new sample of kids at risk for depression because they had mothers with depression and healthy control kids, and assess both behavioral and neural indicators of responsivity, both to negative and to positive information. So they did this, they were remarkably successful at doing so. And they recruited 7 to 10-year-old children who'd never had depression themselves yet, but had mothers with depression. And then they had kids without a family history of depression. And they had them do both behavioral tasks and then tasks in the scanner. And I'll talk a little bit about both of those kinds of tasks. They were 2 different tasks, one they did outside the scanner and one they did inside the scanner. And they looked at both responsivity to loss and gain. So I'll talk about the results here in terms of responses to loss. And a little bit later, I'll talk about the responses to gain, because we saw disruptions in sort of both sides of the equation in these kids. So the behavioral task we did outside the scanner was one designed by Diego Pizzagalli, who's at Harvard University. I think he's spoken at the symposium before. And it's a deceptively simple task, which made it relatively easy to do in kids. So people see a face and there's a mouth, and that you titrate it to make it a very challenging discrimination as to whether the mouth is short or long. And people just say, is the mouth short or long, right? Very simple task. But unbeknownst to them, what happens is on a subset of trials, you give people information about whether or not they were accurate, and they get like a little thing that says like, you won 5 cents, right? It's never false feedback. It's always accurate feedback, but it's only on a subset of trials. And you disproportionately inform them about the results with one type of response versus the other. So we call that the rich response. So it's either saying it's a short mouth or a long mouth. And what very robustly happens, even though people have no sort of explicit or conscious awareness of it, is that they develop a bias in signal detection terms towards the response that is disproportionately rewarded, right? So if I said, I'm going to reward, the short mouth more, on more of the short mouth trials, you get feedback about whether you are accurate or not accurate. And it's a fairly simple task, so people are mostly accurate. And you can really quickly see people develop a bias towards that response. But they cannot tell you, if you debrief them afterwards, that there was any differential information provided about one mouth versus the other. Okay. You can do the same thing in the opposite way, which is you can give them selective information about when they were wrong. Right? So that's the loss condition, right? So you can either focus on when they won or focus on when they were incorrect. And I'm going to start by talking about the incorrect version of the task. And that version, David and Catherine created themselves, because the reward version had already been in the literature, and then they created the loss version, as we call it. And what we did was look at this as a function of both family history of depression and the kid's current depression score at the time of assessment, even though none of these kids met criteria for major depression. And what we saw was that we didn't see differences in performance on the task solely as a function of family history of depression, but we did see differences as a function of how depressed the kid was at the time of assessment. So this is a negative mood score on the child depression inventory. So higher scores are more depression. And then this is the bias on the task, right? And so a more negative value means you are more sensitive to feedback that you have been incorrect and you've lost money, right? And so kids who show more depression are more sensitive to feedback about loss, right? And show more of a bias about that. I'm going to call it away from that information, because the bias is actually away from the choice, the response that is given more information about loss, right? So not as a function of family history, but as a function of how currently depressed the kids were. We then had the kids get in the scanner and do a pretty simple it's not really even a reinforcement learning task because there's no learning. They simply see a question mark and they guess whether if you flipped a card, we call it flipping a card. If you flip a card, is the number going to be above 5 or below 5, right? You could call it like a gambling task, right? So there's no contingencies. You can't learn anything from this task. But if you are correct, right, and this is an aside, when we started this study, everybody told us you can't do money with kids. It's actually not true. Kids understand money just fine. So we decided to do it with candy, and we did it with, and even more funny, we started out with M&Ms, and interestingly, half our kids did not want M&Ms. This was shocking to me as a chocolate lover. I was like, what? But they wanted Skittles. So they could either have, we had a version of the task for M&Ms and we had a version of the task for Skittles. And I will say, we did a behavioral pilot with this where we were trying to like really intensify it. So what we did was we took a bowl of candy and we stuck it in front of the kids and we sat the kids in front of the computer. We did this task and they literally took the candy out of the bowl whenever the kid lost. And we had to stop doing that study because the kids became so disorganized by us taking candy away from them. So just, if you want to know about how to do this with kids, do it more in the abstract or do money, because taking candy from children is not a good idea. So this is how they set it up. They guessed, you know, if it was a 8 or a 9, they got 4 Skittles. If it was a 6 or a 7, they got 2 Skittles. If they lost, you always do half the loss because of how people compute these things. But if they had a 1 or a 2, then they lost 2 Skittles. If it was a 3 or 4, they lost 1 Skittle. And we could look at brain responses to the outcome. In this version of the task, we can't look at cues. We could only look at responses to the outcomes. And so they again looked at this both as a function of whether they were a high risk or a low risk kid, and as a function of their depression at the time of scan. And what they saw was that here we did see differences as a function of risk status. So when we looked at the high risk kids, which are in these kind of arrow things, we saw in the anterior insula and then the ventral putamen, they showed a much stronger deactivation of response when they got information that they lost, right, compared to the kids who were high risk. So not every region of the brain circuit that's involved in responding to emotionally evocative information, but at least in the anterior insula and the ventral putamen, we saw risk group differences. So those studies, and we have several others that we've looked at too, really do suggest that some of these differences, both behaviorally and in terms of brain responses in systems involved in negative valence, emerge very early in life. They are disrupted in kids who have early onset depression, and to some extent, at least, they're disrupted in kids who are at risk for depression and who do not yet have full-blown clinical depression. Again, suggesting that some of these differences are arising very early in life, you know, in at least some cases prior to the onset of full-blown clinical disorder. We then wanted to know, well, how much might this relate to having difficulties with emotion regulation? Normatively, right, we know that emotion regulation develops across the course of development. You do not expect a 5-year-old to have high levels of emotion regulation as a normative thing, right? It's gonna emerge normatively. But nonetheless, there's still a great deal of variation in emotion regulatory capabilities, even amongst 5, 6, 7, and 8-year-olds, right? So there's still room to look at whether children with depression might be having greater difficulty with emotion regulation. And this is, you know, not the easiest to study in kids. You can do like frustration tasks where you have, you know, kids engage in something that's very frustrating and you videotape them and code them. If we do that, we do see indeed increases in sort of, you know, lack of evidence of engaging in emotion regulatory abilities in kids who have early onset depression. We certainly see that parents rate their children with depression as having a lower emotion regulation skills and greater emotional lability, particularly negative emotional lability. But many of those parents also have their own mood and challenges. So sometimes it's a little bit hard to know, are they rating, you know, how much of is their own mood challenges biasing their rating of kids? So we were kind of struggling to think about what could we do with neuroimaging. And Ian Gottlieb, who's at Stanford, was a collaborator of ours on the grant at this time, and he suggested that we try a task that he had been using. And he had a sample of older kids at risk for depression by virtue of having moms with depression. And he said, why don't you try the task that we're trying? So we did the task he was doing, which was pretty straightforward. Not the most well-controlled study I've ever done, but we had them do a baseline rest scan. And then we had them watch a film clip that was designed to elicit a negative mood, right? It was, I forget the name of the clip now, but it was a very sad clip where a kid gets bit by bees and gets very sick. So we have them do that. Then we give them a minute to ponder that negative mood, and we're scanning during this whole time, right? And we looked at, and this was work that David Pagliaccio spearheaded, and we looked at the degree to which kids were able to activate regions involved in emotion regulation during these periods where they're getting essentially a negative mood induced, right? And then we did a positive mood induction repair afterwards so that kids didn't leave the scanning session in a negative mood. So what David did was then look at differences in brain activity during that mood induction period compared to the the pre-mood induction rest, and then looked at those in relationship to the severity of preschool depression. And what he found was that there was less of an increase in activity in the dorsolateral prefrontal cortex, a region sort of canonically associated with at least certain forms of emotion regulation, amongst children who had higher depression severity, coupled with greater activity in regions like the amygdala in children with higher depression severity at the time of scan. I don't think this is by any means proof that they're having difficulties with emotion regulation, because we certainly can't tease apart whether it's greater, say, amygdala responsivity, making it more difficult to regulate emotion, or difficulties regulating emotion, allowing the amygdala to show greater responsivity. But it's at least consistent with there being some sort of disruption in emotion regulation very early on in life in kids who have a history of preschool depression. Okay. Then we wanted to look at connectivity, because we know that the brain, none of the brain regions that we're interested in operate in isolation. And even when you think of them in their own sort of local circuits communicate with each other and work together to give rise to more complex human behaviors. And in particular, there was a lot of work arising in the adult literature looking at connectivity, for example, between amygdala and sort of emotion regulation regions of amygdala and the ventromedial prefrontal cortex, and suggesting that adults with depression show disruptions in those patterns of connectivity. So we wanted to look at that in our kids as well. And this was work spearheaded by Catherine. So what she did was look at connectivity between the amygdala and what we're calling the negative network, meaning the other regions of the brain that tend to get activated when kids are responding to, So when you look at amygdala connectivity, you see two patterns in the brain. So you see connectivity between the amygdala and regions like the insula, and to some extent the ventromedial prefrontal cortex, that all seem to be going up together when kids are being shown emotionally evocative pictures, especially negative pictures. But when you see increased activity in the amygdala and these other regions, you tend to see decreased activity in the dorsolateral prefrontal cortex, the dorsal parietal cortex, the dorsal anterior cingulate cortex. And when you have people just rest quietly in the scanner and you look at spontaneous fluctuations in the bold response, right, which is an indirect measure of brain activity, what you tend to see is some people call like a seesaw relationship or a push-pull relationship, where when activity is going up in the amygdala, it tends to be down in these prefrontal, and dorsal parietal regions, right? So if you were to compute the connectivity between the amygdala and these cognitive control regions, that connectivity, that correlation really would be negative, right? So that would be the typical pattern that you would see. We see this both in children and in adults. And what we saw when we looked at kids with depression, so we looked at healthy control kids who had a mom with depression but didn't have depression themselves, kids with depression but who didn't have a mom with depression, and kids who sort of had a double whammy, both having a mom with depression and experiencing depression themselves. And what you can see is that across the board, more so for here, but it was very closely trend level here, a reduction in that negative correlation, right? So normatively, you see this anti-correlation, this negative correlation that people think reflects sort of top-down control over emotion processing regions. And the degree of that anti-correlation is reduced in kids with depression or at risk for depression. And if you look at individual differences at the time of scan, so this is a parent report measure of emotion dysregulation and sadness dysregulation in particular. So higher scores are parents reporting greater dysregulation around sad emotions in their children. You see that the greater the parent report of emotion dysregulation, the less kids are showing this anti or negative correlation between amygdala regions and these kind of prefrontal and parietal putative cognitive control regions, right? So starting to see some evidence also of disrupted kind of, in theory, communication between these two brain systems. Now, Mike Gaffrey was a postdoc with us that went on to become a faculty member in our Department of Psychiatry and has since moved to Duke University and is an assistant professor in the Department of Psychology there. And he really spearheaded pushing the envelope and doing imaging with 4, 5, and 6-year-olds themselves. So most of the studies I showed you, right, we recruited the kiddos when they were 4, 5, and 6, but we didn't start scanning till 8 years old. That's challenging enough. But what Mike did was decide, let's really make it hard for ourselves. Let's scan 4, 5, and 6-year-olds. And he was remarkably successful at this. He put a great deal of effort into this. He developed all kinds of great systems for doing this. So I still consider him to be sort of one of the world's experts in doing this successfully. And what he did was look at amygdala functional connectivity and emotion regulation in these 4, 5, and 6-year-olds themselves, right? So going even earlier than we had been able to look at. And what he saw was more evidence of a disruption in the connectivity between the amygdala and the medial prefrontal cortex rather than the dorsolateral prefrontal cortex and dorsal parietal cortex in these even younger kids. And when you look at, these are parent-reported emotion regulation measures in the kids. So in this age range, it is more normative for there to be a positive association between the amygdala and the ventral medial prefrontal cortex. There's some literature suggesting that the direction of this functional connectivity actually switches around puberty. Dylan G. and BJ Casey have some cross-sectional studies of development that suggest that it switches at around puberty, but in these younger kids, you tend to see a positive correlation. And what he saw here was more of a positive association amongst those children whose parents reported them as having better emotion regulation, and more of a negative correlation in the kids whose parents reported them as having poor emotion regulation. And all of his kids were recruited because they had depression, right? So he recruited specifically kids who had early onset depression. So from 2 different kinds of studies, both in preschoolers themselves and in school age kids who have a history of depression, we already see evidence of disruptions in functional connectivity between regions like the amygdala that are thought to respond to salient information in the environment and regions that might be involved in attentional control or top-down regulation. Okay. Then we turn to looking at positive valence systems, right? Because, it may also be important to think about the balance of emotional experiences between negative and positive. And I think particularly in the last 10 years, there's just been a huge amount of interest in depression on positive valence systems. A lot of work in adults suggesting that there are like in meta-analytic work, relatively focal reductions in the activity of brain regions that normally respond to rewarding or pleasurable information. So this was a meta-analysis by Karen et al. That looked at studies of reward processing in adults with depression, and they systematically saw differences in the ventral striatum, specifically reductions in bold responses in the ventral striatum amongst adults with depression. But even before this, people like Erica Forbes and Ron Dahl many years ago were actually already starting to talk about disruptions in the neural systems associated with positive affect in understanding child and adolescent depression, right? And Erica was really one of the first people to start to look at this in adolescent and child depression, finding evidence indeed that kids with pediatric depression showed reduction in activity in subcortical brain regions that typically do show increased bold responses to kind of information about having either positive social outcomes or positive monetary outcomes. She's done some of both. So we wanted to look at this in our kids, and we kind of wanted to look at it in multiple different ways. So we wanted to look at how does the kind of timing of depression, when it's occurring, how long it's lasted, relate to the degree of disruption in these positive valence systems. So we turned again to the study I told you about earlier, the preschool depression study. And this is just a little bit more of a schematic. So, we started recruiting these kids in preschool. We've been following them through adolescence. We have these multiple scan waves. And we have measures of depression starting in preschool, and we have them coincident with scanning as well. So we can look at, if you were looking at, in this case, we started when the kids turned, it was like about 14, 15, we were able to do a bit more complex tasks in the scanner. So we added in a task similar to the Skittles one that we had done earlier, where, kids made predictions about whether the number was going to be greater or lower. Then I'll actually show you a figure of that in a minute, but greater or lower. And we were actually able to look at both cues about what they might win or lose and the actual responses to winning or losing. So a little bit more complex task as the kids got older. We did both ERPs and fMRI measures of this kind of reward processing. I'm only going to have time to talk about the fMRI for this part here. And then we looked, and this was the exact design of the task. So they guessed, they got a cue as to whether it was likely that they were going to get nothing. It could be either they were likely to lose, likely to win, and then they got their actual outcome. So we can separately look at brain activity to these cues and to the outcomes, and try to look at anticipation versus kind of receipt of reward. There we go. Oh, I forgot I put this in here. Okay. Catherine then went back and looked at the ERP data to the same exact task, right? So I'll start by showing you the ERP data. So she looked at responses to wins versus losses, which is a ERP component, used to be called the feedback negativity. Now everybody calls it the reward positivity. It's the exact same thing. People just decided we're going to have a new name now. Go figure. And because we were good little ERP doobies, we plotted negative going voltages upwards rather than downwards. And so what happens is when people get feedback that they've lost, right, they show a negative going voltage. And when they get feedback that they won, they show less of that negative going voltage. And if you look at the difference, that's either the feedback negativity or the reward positivity. And so what Catherine did was look pretty systematically at this in relationship to depression kind of across the kids' lifespan with us. If you look, I realize this is maybe more complex, you'll have to take my word for it. So she looked at kids who had high levels of preschool depression versus low levels of preschool depression. And the basic finding was that we saw reductions in these responses to reward-related outcomes amongst the kids who had high levels of preschool depression. And we tried to look separately at preschool, school age, and adolescent depression. And I say tried because they're highly correlated, so it was pretty difficult to disentangle them. The strongest signal was coming from the preschool depression severity. And we've pretty much, I mean, spoiler alert, we pretty much always see that. I think that may be an artifact of our sample because they were recruited for having high levels of preschool depression severity, but we definitely see the strongest signals in terms of all of our neural and behavioral outcomes for the most part in how severe their preschool depression severity is. So it may be an artifact of our sample. It may also be that that's a really critical period for kids to be experiencing depression because so much learning about attachment relationships and emotion processing and emotion control is happening during that time period. And being pretty severely depressed is going to disrupt all of those relationships, but we can't rule out that it's a sample specific artifact. Okay, so those are just the differences. Okay, so now let me move to the fMRI. So this is first just the group level responses to the outcomes, just to kind of illustrate that the task worked. And we see greater activity in a variety of brain regions that are known to respond to positive versus negative outcomes. You know, both the dorsal and the ventral striatum. We see it in the, the kind of rostral anterior cingulate as well. We do see some in the visual cortex. And this is the outcome phase. If you look at the Q phase, you see a bit more focal activity, but it is some of the same regions, right? The dorsal and the ventral striatum, and also in the visual cortex. So this is just the group level, the task worked, we get activation in the regions we expected. And then my wonderful graduate student, Brent, went and systematically looked at, well, how does depression severity at the preschool age, the school age, the adolescent age, current depression severity, relate to responses in sort of each of the regions that we typically think of being associated with a sort of reward processing network. So different regions of the striatum, the dorsal anterior cingulate, the insula, and to the circuit as a whole. And do we see any evidence of sort of differential relationships there? And what he saw was that if you looked at current depression, right? So this is kind of a whole cortical striatal network that responds to wins versus losses. And that's made up of the caudate, the putamen, the nucleus accumbens, the anterior insula, the dorsal anterior cingulate, and the rostral anterior cingulate. Saw some relationships, but nothing really to write home about. Primarily, it was in the nucleus accumbens that was the strongest relationship, where the more severe the current depression, the less activity kids showed in response to wins versus losses. And when we broke it down, it was really about the responses to wins, not about the responses to losses. Then he looked at cumulative depression severity. So what was their cumulative depression severity across preschool and adolescence? And now you see much more robust relationships across this whole cortical striatal circuit. You see it in this kind of aggregate measure, and then you see it with almost every one of the individual regions. And then if you look at preschool severity, right, again, you see stronger relationships than you do for current severity even, right? You see it with the whole cortical striatal network, caudate, putamen. You actually don't see it with the nucleus accumbens, but you do see it with the dorsal and the rostral anterior cingulate cortex. And then if you look at school age, you see something a little bit in between. And if you look at adolescence, right, you see kind of the circuit as a whole, but much more focally the striatal regions. And the potentially interesting interpretation here, which we can't prove, but is where we want to look at this in longitudinal studies where we measure brain activation even earlier, is if you experience depression very early in the course of life, right, it may have these sort of propagating effects, right, where it impacts activity across a much broader circuit over time. If it's blunting your learning about reward information because you're not having a very strong hedonic response, and that kind of cumulative depression severity, and particularly early on, may be much more broadly associated with disruptions in the sort of whole circuit that responds to rewards and losses. Whereas current depression severity, which might be occurring for some kids for the first time, right, might be more focally related to disruptions in the brain regions thought to be kind of most centrally associated with the hedonic response to information about rewards. We can't answer that question from these data because we only image these kids as adolescents. But in other studies where we're starting imaging them early on, we can look at basically whether there's sort of an accumulating spreading of disruption in these circuits the longer kids have depression, which would be one hypothesis. This just kind of gives you a sense of the magnitude of the relationship. So this is just cumulative depression severity and the kind of activity in the whole cortical circuit. So it's the same data I just showed you, but I always like to show you that there's not gigantic outliers driving it. Okay, so evidence in kids, preschoolers through adolescence, that this early onset depression really is associated with disruptions in all of these systems that we see disruptions in adults with depression. We're still trying to get our handle on how early things start. So one of the things we started doing with Joan is started moving towards treatment of kids with early onset depression. So we ran another study that we call the preschool depression treatment study. I'll tell you a little bit about it because I'm actually going to show you the results of the study too. And again, this is a treatment that Joan developed, so I don't get any credit for developing the treatment. So she modified preschool PCIT, which is parent-child interaction therapy, already really well validated for kids with disruptive disorders. There's components that focus on parenting skills, components that focus on the child and their interaction. She developed a whole additional module called emotion development that was all about helping moms to basically help kids learn how to regulate their emotions, both to upregulate positive emotions, because in depressed children, you see a real lack of joy, and also helping them to down-regulate their negative emotions, deal with frustration and so on. So the design of the study was we brought them in at baseline and we did both ERP and fMRI assessments, and the ERP assessments focused on being able to look at responses to rewards and losses. These were 4, 5, and 6 and 7-year-olds, so we had to keep it pretty simple. We couldn't do the real fancy tasks. Then they went in and did treatment for 10 weeks. The standard PCIT. We did an interval assessment here of the same ERP task. Then they went and did 8 weeks of the emotion development component. And then we did a post assessment with ERP. So let me first show you the baseline ERP, and then I can show you what happened with over the course of treatment. I just said that right. The task we use, very simple, well-validated task in the ERP literature, developed by Greg Hychak. He was our collaborator for this study. Kids just pick a door and they win or they lose. And we did money here, points that translated to toys. That worked just fine. Kids totally got points and toys. We had a whole store set up for them so they could see ahead of time. They could, pick out prizes ahead of time. So we can look at it, but here you really can only look at responses to the feedback period is really the best you can get out of this kind of paradigm. Okay, so what do we see before kids start treatment? So here is a scalp map for a healthy child. The kind of warmer colors indicate essentially stronger responses. I will say that in kids, we see responses. Normally in adults, the response to this feedback negativity, the reward positivity is more anterior. You're more likely to see it in FC. We saw it much more strongly in kids in CZ and PZ, which is more of a posterior distribution. We've seen that in a bunch of studies now. We don't know exactly why that is, whether it's scalp shape or, fat composition in the head, or whether it's actually being processed differently in terms of neural systems, but we see a much more posterior distribution. Here are kids with preschool onset depression, and you can already see that the colors look much cooler back here. And if you do a direct comparison, this is the mean amplitude at PZ in our healthy kids versus the kids with depression, and we see a significant reduction in the amplitude of the response to rewards versus losses in our kids with depression prior to treatment. So then we go and we do treatment, or Joan did treatment, and it uses a bug in the ear approach, which is kind of a coaching approach. So our labs are set up where we have a one-way rooms with one-way mirrors, and we have a therapist who stands in the middle, and it was mostly moms, not by design, but just by happenstance. And they wear a microphone in the ear. They have structured tasks that they're supposed to do with the kids. The therapist coaches the parent while they're in the room with the kid. They're supposed to go home and engage in activities at home. And so they get this whole coaching approach. And really it's focused on emotional reactivity. Like I said, both kind of upregulating joy. So teaching parents how to help kids like savor positive things and get excited, but also helping them to regulate their emotions when they get upset or frustrated. So it's really kind of capitalizing on the parent as an important component of that emotion regulation dynamic for kids. And we, like I said, we use structured emotionally evocative tasks in the session. Okay. This is just an example of what our setup looks like. So she's the therapist. Here's an example mom and kid who gave permission, and they're doing these tasks. This treatment works for kids with depression. So this is kids so this is the percentage of kids who still meet criteria for major depression post-treatment. And in red is the waitlist control group. We then cycled back and offered treatment to all of the kids in the waitlist control. And this is the kids who get PCIT. So there's a reduction in the number of kids who experience depression. The exact opposite is more kids remit from depression in the PCIT. And also, this is a dimensional measure called the preschool feelings checklist, filled out by a blind rater that doesn't know which treatment the kids are in, because parents obviously know what treatment kids are in, right? And we see a greater reduction amongst the kids in the PCIT treatment. We looked at a whole bunch of other outcomes. Kids' global functioning, again, assessed by a blind rater, gets better post-treatment in everybody, but it gets more better in the kids in PCIT. We look at, in this case, parent-rated emotion regulation. So take that with a grain of salt, because parents do know what treatment they're in, but we see a greater improvement in the kids in PCIT. And then this is negative lability, again, rated by the parents. We do see a reduction in negative lability amongst the kids with depression. A couple things to tell you about this. We followed them out for a year, and the gains are maintained in the kids out to a year. Parents actually experienced significant reduction in their own depression during the course of the study. That was not as robustly maintained as the kids' depression. We also looked at the degree to which parents continued to engage in the parenting skills they were taught. And they do, to some extent. There is a bit of falling back. And so we're thinking that booster sessions might be something that would be really helpful, because these are challenging parenting skills. We also looked at breaking it down by module as to when the treatment changes were occurring. And we definitely saw a disproportionate improvement in the kids' depression from the assessment prior to the PCIT versus after. We did see some improvements in the other modules too, but we saw the biggest improvement from pre-post emotion development module as well. We now need to do some dismantling studies, and also like thinking about, the problem is it's a fixed order, right? And it's a little hard to know, is it just time, like more treatment, you get better, or is it really something specific about the emotion development module? So that's something we still need to work on. Okay. And their anhedonia gets better, which was of particular interest. So then a question for us was, well, what happens in terms of the brain responses to rewards? And maybe not surprisingly, they improved post-treatment. So we saw a significant increase in responses to the rewarding outcomes amongst the kids with depression who were in PCIT versus not. So this was the final post-treatment assessment. We then went back and looked at the ERP assessment that happened prior to the onset of the emotion development module. And we did not see a difference there versus pre-treatment. The differences all occurred from the midpoint to the post-treatment, which was during the course of emotion development. I will say we also looked, the individual differences in response to reward pre-treatment did not predict who benefited the most from treatment. What did predict is we actually had done another task that was kind of almost like an implicit emotion regulation task with kids. We showed them pictures. They just had to respond to the pictures, but pictures could be happy or sad. And we were looking at, do they show a what's called a late positive potential? Do they show a modulation of this component? We did not see a change in that as a function of treatment, but the kids who showed the greatest increase in the LPP at baseline to positive pictures were those kids who were most likely to benefit from PCIT. So we don't know if that means they just had maybe a little bit better emotion regulatory abilities at baseline, and therefore were better able to be scaffolded, or, better other aspects of emotion processing systems. But the reward responsivity did not predict. Okay, I'm almost out of time, aren't I? 5 minutes, okay. I wanna show, let me skip over the, I'm going to skip over this. I want to show, okay. I've never gotten to talk about all of this in one place before, so it's like super exciting. So I remember I promised you that in the depression risk study, we looked at reward as well as loss responses. So I just wanted to circle back to that. So same study I told you about before that Catherine and David did. Anyway, I'll get back here. So now we have the version of the task where it was selectively about rewards and not losses. And what they found was a kind of a parallel finding to the negative responsivity. So greater mood depression. So now in this case, you want to be biased towards the reward response, right? Because that's good. You know, if you're responding to the reward. And what they found was that the kids who had the most severe negative mood showed, if you recall before, the greatest bias about the losses and the least bias about the rewards, right? Didn't differ as a function of family history of depression. This was really more about the kids' current depression history. And then again, we had the scanning task, and they looked at the responses to rewards. And what they saw here, so this is again the anterior insula. Before I showed you this finding about greater deactivation in the high-risk kids to loss, that was coupled with less activation in the high-risk kids in the anterior insula and in the ventral putamen to rewards. So healthy kids normally show an increase in activity in these regions. That's what you'd expect in adults too. And the kids at risk for depression did not show that, right? So again, some evidence of disruption in the positive valence system amongst these kids at risk for depression. I wanted to show this one last study actually because I thought it related in interesting ways to the work that Bo Lee presented. This is back to the work of Mike Gaffrey, who, in addition to doing resting state connectivity in little tykes, managed to get them to do a task in the scanner, which I, again, have to give him so much kudos for. So this was the same study. He recruited a bunch of kids with early signs and symptoms of depression. And then he created a preschooler specific version of this reward task. So again, it was about M&Ms or Skittles. We super simplified it. So now the task was, is the figure you're going to see going to be bigger than you or smaller than you? So this was them, this was bigger, and this was smaller. And we ordered a custom response box from Current Direct Designs, which makes our scanner button boxes. So there was just a single response for the kids. We Velcroed it to them so it wouldn't go away. And they just pushed the button. So we only got one response, which was a downside, but we were finding that kids had a really hard time not doing this if we had them do more than one button. So by just doing one button, we were able to get them to, well, Mike was able to get them to keep their head straight up. And so he did this task in the scanner with them. He also did a child version of the Trier social stress test and collected cortisol. Like this was a study. Okay, so first, let me show you what we saw in terms of brain responses in these little kids. So the three regions in particular that showed the strongest responses were the right caudate and the left caudate and the left amygdala. And, you know, not the cleanest brain waves we've ever seen. But again, this is pretty challenging to do this in this age range. So the blue ones are responses to gain, red responses to loss, green, the neutral. And I think there's two interesting things here. So in the right and the left caudate, we really do see something analogous to what we see in adults, which is greater responsivity to gains than losses. Neutral was pretty similar to gains in the kids. But interestingly, in the amygdala, right, what we see is they respond to salient things, right? They respond to both positive and negative more so than neutral, right? So this was really interesting. We did not actually go into this anticipating that we were going to see this amygdala response. It fits with some of the other literature. I think it fits with some of the work that Dr. Lee was presenting. Then he went and said, okay, how do responses in these brain regions relate to depression severity and cortisol responses to this Trier social stress test? So he computed area under the curve for cortisol. He had multiple assessments and he computed sort of the magnitude of the overall response. And first, in these little preschoolers with depression, you see something that is sometimes seen in adults, which is greater depression severity in the kids is associated with a stronger cortisol response to this social stressor. So we see positive correlation between cortisol area under the curve and depression severity. Then he saw that amygdala response was related to cortisol responses. So this is greater cortisol response, a stronger depression reactivity right here. you actually see less responsivity of the amygdala to these salient things. I think I'm saying that right. Oh yeah, because it's more negative. Then he looked at amygdala in response to depression, and you see the same thing, that the kids with depression are showing kind of a blunted response of the amygdala to information about wins and losses. And then if he did at least a cross-sectional mediation, I'll say it's a model of the plausibility of mediation, right? Because it's cross-sectional data, is he saw that this blunted amygdala response mediated the relationship between stress responsivity and depression severity. So greater stress responsivity, more blunted amygdala response to salient outcomes, and greater depression severity. Now, in a lot of the adult literature, you might have predicted a greater response of the amygdala, at least to the loss information. That's not what he saw. He saw more of a blunted response in the amygdala. And notably, in these little tykes at 5, he did not see that depression severity related to the caudate response, right? That was not related to depression at this age. But he did see some evidence that these sort of amygdala, we've been thinking of them as sort of salient responses, did seem to potentially play a role in the association between greater stress responsivity and depression in these kids. That was across. So the amygdala didn't show a differential response to gains and losses. It showed a greater response to both compared to neutral. So this is responsivity to salient outcomes versus no outcome, basically, right? So it didn't show an emotion difference. Okay, so now I will finally summarize. So we do see in our hands, and I think other people are seeing this too, that kids at risk for depression or with early depression do show brain differences that are analogous to what has been seen in adults with depression. We see increased activity, at least in some studies, in regions involved in negative valence systems. We see decreased activity in brain regions involved in emotion regulation, and we certainly see a lot of parent and teacher and observational measures indicating emotion dysregulation. We see disruptions in the connectivity between these emotion regulation regions and emotion responsivity regions, and we see reduction in brain activity in regions that respond to positive outcomes or positive valence. We see it fairly early on in life, but we see it most strongly in kids with like a long cumulative history of depression or with particularly severe preschool onset depression. Is this a scar? Is this risk? It seems like it's not just a scar, because we do see some of it in kids who've not yet at least had a full clinical diagnosis of depression, who are at risk for depression. But it certainly is worse with earlier onset and cumulative depression severity. So there may be both a role for risk processes and some impacts or scars of cumulative depression. We see at least some evidence that it can improve with treatment early on. And if Joan Luby were here, her mantra is, I really believe kids' brains are more plastic early on. And so we should be doing interventions early. I do think that's true, right? And then, I do think we probably need to be thinking about more sustained interventions from kids, because I don't really believe that it's kind of one and done. These kids are probably going to need some ongoing support, but a lot of these kids end up having a lot of difficulty with peers, a lot of difficulties in school. They're, again, missing out on what I think of as normative experiences that are important for, positive emotion development and effective emotion regulation. So to the extent that we can help them be able to better engage in those processes and build on them, I think the better off these kids are going to be, even if they continue to experience some difficulties throughout their life. I think we still need to better understand mechanisms, right? We haven't, for example, really in our hands looked at genetics. I could give you in a 2-hour talk about early environments and poverty and how much they relate to depression and disruptions in a lot of these brain systems, but I won't do that to you today. So we think there's really interesting animal models that help articulate what some of those mechanisms might be. So we certainly think that's playing a role as well. But again, early intervention, early treatment, broader treatments, we have more kids coming into the clinic than we can help, right? Which is very sad. We have studies of preschool onset suicidal ideation going on now. So these kids also articulated at the time, which was a shocking level of suicidal ideation. Joan could give you a whole other talk on that too. We actually got a supplement to the grant because we were just so shocked by how much suicidal ideation we were hearing. And we did a bunch of measures of understanding of death and dying. And it turned out that our suicidal preschoolers understood concepts of death better than our non. So it wasn't that they didn't understand, and they were doing things like putting bags over their head, running out into traffic, saying they were going to put daddy's tie around their head. So these are actions that could actually harm you, right? So there's clearly a greater need for early intervention with these kids. So I'll stop there. Thank you for letting me say it all at one time. This is nerve-wracking to know that you've been preparing questions.
Sure. Hi, Dr. Barch.
Hi,
Thank you for your wonderful talk.
thank you.
Sure. My name is Camille Williams. I am a third-year doctoral student. I work with Dr. Simon Goldberg and Dr. Sarah Short in the Center for Healthy Minds. I am particularly interested in parent-child interactions, actually, and empathy development in early childhood. I will start with a question here. You discuss in your work that children with depression often have issues with reparative pro-social behaviors, and that this is often an issue of the child feeling overwhelmed by guilt. What I'm curious about is, how do you discern instances in which the child may feel too little guilt, given that there's high comorbidity with ODD and conduct disorders? Yeah, and in cases like that, how might the PCIT-ED intervention look different?
Yeah, that's a great question. So first, I'll give a shout out to Rose Donahue, who's another one of our fabulous team of folks, and she is particularly the reparative, pro-social, and antisocial behavior person of our team. And she has a lot of really interesting experimental paradigms where she is looking at these kinds of things. Can you look at too little guilt versus too much guilt? What kids say is very different under those different situations, right? If you're, talking to a parent about a situation that happened, often in this age range, right, you're either getting your information from the parent or a teacher, or you're having them do tasks in the lab and you can see what's going on. So she has this one task, which has been you're going to think we're horrible people when I describe this task to you. But so what she does is she has sometimes it's her, but a confederate and the child go in a room and they're talking, and another experimenter comes in, and the experimenter has a picture and shows the kid and the experimenter and says, this is a picture of my family. It's really important to me. Like, oh, isn't this, I love it, whatever. Sets it down on the table and leaves the room, says, oh, I have to go do something. That experimenter leaves the room. The other experimenter says to the kid, we should rip this picture up.
Nice.
And then proceeds to spend time trying to convince this kid to rip up the picture. And you can look at, there are kids who are like, sure, boom, right? And then there are kids who are really resistant, and then you can look at what they do when the experimenter comes back in, and then they do a very extensive, positive reframe. But she has been using that to look at the kids who seem to have no problems, right? Just being like, I don't care if it bothers them, I'm going to rip it up, to the kids who are really torn. And even because then you can look at the excessive reparative and guilt, you can look at, well, what happens now when the other experimenter comes back and it's like, oh no, I got like 10 copies of those. It's no big deal, right? And the person told you to do it. The kids who still can't let go of the guilt, right? Like it takes them a while to be convinced. they do it because they're often, the experimenters telling them to do it, they want to follow these instructions, but they have this really excessive guilt. So that experimental parent, she coded for this stuff and found that it predicts callous and unemotional traits like in adolescence. Like it's really a pretty powerful predictor. So that's not the easiest thing to do in a clinical setting, although she has actually argued that you could come up with a little clinical vignette of that to kind of really tap into it. But that's one of the ways she's tried to get at it.
Thank you.
Sure.
Hi, I'm Danielle. I'm a second year graduate student in neuroscience. And I was really fascinated by reading about the clinical trials of the PCIT intervention. It was very uplifting to read that it's very effective, especially even how sad it is to read about preschool age depression. So my question is, the original PCIT was designed not with depression in mind, but with behavioral issues in mind. And since it was initially developed in the 70s, it wasn't developed with since then, there have been a lot of advancements in our understandings of the neurobiological mechanisms of depression. So given that, I'm wondering, how do you see the PCIT intervention protocols growing and changing to reflect the new goal of depression intervention and the new understandings of neural mechanisms?
Yeah, great question. So you're absolutely right. PCIT itself, designed long ago, was not at all designed with depression in mind, right? And in fact, it hadn't really been tested to see whether it was effective for depression. I wish I didn't pull up the paper, but we have this paper, this kind of dismantling paper, and I'm trying to remember now, we definitely saw improvements in ODD behaviors during the parent and child oriented part. And I think we also saw continued improvement in the emotion development part. And we did not see a lot of improvement in depression during the PCIT part and more improvement in depression in that. So I would say the ED was specifically that, like it was designed with what we know about depression and thinking about behavioral activation, emotion regulation, positive valence systems. Like when Joan designed it, not me, she was really thinking about, here's what all the data say about the behavioral and the sort of neural systems that are dysregulated in these kids with depression. We've talked a lot about next steps. I mean, we've talked about probably the need for boosters. We've also been talking about broader dissemination and like how short could it go, right? Because I mean, having the whole thing took 18 weeks when we did it, and we paid people to come in and we provided childcare and like we did everything we could to support these families coming in. And she has a clinic where she offers it clinically, but that's not really accessible to a lot of families. So she actually did start another trial, and then COVID interrupted, in one of the low resource school districts in town, doing it after school with kids who've been identified by teachers. We didn't end up getting a very big N. We saw hints that it was effective. And they're now working on a really short, like 6-week version that could be pushed out into school. So I think thinking about how quickly we can get change is definitely something we need to be thinking about in terms of the treatment. In theory, we could be thinking even younger, although I think it's going to be hard to go much younger than 4 or 5 for really intensive treatment. I mean, Richie and I were just talking about kind of some interesting ideas about, could you do things even younger focused on the parents? I hate to say, I always say moms because the moms were the ones who showed up for that study, but whoever the caretakers are, either to explicitly train them early on if they know their kids are at risk, or to do, mindfulness interventions or things that might end up being useful in terms of, I just do think there's something really critical about trying to make sure the kid is having as much normative experience developmentally as possible so they can sort of benefit from all of those things that, we don't know for sure that there are like full on critical periods in emotion development the way we know about it, like early attachment, but one would certainly hypothesize that those are particularly sensitive periods. Does that answer your question?
Yeah, that's fascinating. Thank you.
So I'm Will Booten. I'm a 4th year in the clinical psychology program here. So sort of building off of the question that Danielle had, we know that a lot of our currently existing interventions weren't developed necessarily with these specific neurobiological mechanisms in mind. And even though I know you described how PCIT was sort of built with some of these, or at least the ED component was built with a lot of these in there. I also know that in your research, you highlight a lot of the positive affect dysfunctions. So I'm curious if you were to adapt an existing intervention to better target those mechanisms, or if you were to even develop a novel intervention, what would that novel intervention consist of, or what particular intervention?
So a really easy question.
If you were to pick maybe a currently existing intervention to modify, which one would you start with?
Well, it would all depend on the age range, right? So my answer would be very different depending on the age range, right? So for example, if you think about CBT, there's a lot of things you could do to add in like savoring components, right? But I don't think you're going to do that with a 4-year-old, right? I don't think it's really accessible to them. So I think that, in kids, right, like what we're trying to do with the ED, where you harness the parent, because I think of it as like, again, I'm thinking about typical development. We don't expect kids' development to occur in a vacuum, right? So we're thinking about them existing either in some sort of caretaking unit, teachers, other people that are impinging on their life. So I think a lot about how do you harness those people who should be sort of forming that network for them. We do not at all think about going invasive in kids, right? We don't think about medications. We don't think about TMS, or, because there certainly are things you could think about doing in older kids that would target those neural systems specifically. There's been interesting work trying to get going in adults, looking at kappa opioid systems, which are thought to maybe be relevant for the hedonic response. We just wouldn't do any of that in kids, right? So we are a little limited in what we can do with the kids. So I'm not sure I have great ideas for additional things with little kids, other than also thinking about, doing things in the preschool, right? And doing things in the school, because the home setting is only one of the settings in which the kids are operating. And in kids who spend a lot of time in preschool or school, right, those people are also serving as emotion regulators for the kids as well, and could be in theory harnessed to help target these systems, right? And if you think about, if you've ever spent any time in preschool, like one of the tasks we use in our kids is the bubbles task, right? You blow bubbles. And if you take a healthy kid who's not depressed, like, oh, they love it. They're running around, they're popping. You take a depressed kid and they're like, so a teacher could easily detect that too, and be thinking about sort of interventions with the kid about helping them to kind of attend to the positive emotion, upregulate the positive emotion in the same way that we teach parents to do in PCIT ED. If we get older, I think we're going to start to potentially see, I don't want to call them invasive necessarily. I still think medication is never going to be the first line choice, except for some really severe depressed kids. But if we start to find that some of these like stimulation techniques are safe and non-invasive for adolescents who have pretty severe depression, I think we have to be weighing the risk benefit. Even though maybe there's added risk, I think there's so much risk associated with severe depression, again, during these critical developmental periods, that we have to kind of weigh that. I wish I had better ideas.
Let's go to the audience. Let me come back to you.
Okay.
Hey everyone, I'm Katie. I'm a graduate student at UW-Madison, and I actually did work with Deanna Barch before coming to graduate school, so it's always nice to see your face again. And I worked with her on schizophrenia research, so I love hearing all the other kind of research that you've been doing. But you touched a lot on like the importance of boosters and intervention in the work that you've been doing with Joan. And I'm wondering if you've thought about like predictive measurements and how to use like the research that you have been collecting and data that you've been collecting in like machine learning techniques or some other predictive statistics to understand like where and how to give that booster in particular, because you talked a lot about feasibility and like how to actually implement this in like the school systems, and maybe using like the instruments that you've collected to understand how to best operate that, and maybe like what reward or emotion regulation is most important and when in childhood development.
Yeah, those are great questions. So We haven't thought about it from a machine learning perspective yet because the samples we have available for this kind of stuff are just not big enough to do that. But where we have started to think about is using mobile sensing and ecological momentary assessment to think about, because to me, that could be a real way to target when boosters are needed, because it's a way to be assessing kind of what's going on with the kid and what's going on with the dyad. So we have a study ongoing now in those same PCIT kids that was more focused on the suicidal ideation, where we actually got them to agree to do, it wasn't every day, it was every week for a year, to do assessments. And what the idea would be to look at, and we have like parent-child conflict and peer conflict and their mood. And there are definitely people in the adult literature starting to use these techniques to say, can we see early precursors of relapse, right? Or where you might, so you could imagine, right? Maybe you need to do boosters after stressful life events, right? Like they, or where you start to see, depression levels creeping up, right? There could be ways you could get information from that about when you might best think about boosters if you wanted to do more personalized, like triggering of that, as opposed to like everybody gets it at 12 months, right? Maybe you could say, well, you get it when, for at least a month, you've seen an elevation of X on these scores, or you're seeing increased, parent-child conflict or child-peer conflict, something like that. Yeah, so great idea.
Thanks, Deanna.
Hi, Dr. Barch. Thanks
Hi.
so much for a really fascinating talk. My name's Rachel. I'm a graduate student here at UW-Madison in the Neuroscience training program. I was especially fascinated by the last study you talked about, where you sort of saw this relationship between increased cortisol and sort of blunted amygdala activity. And you kind of briefly mentioned that's sort of the opposite of what we might expect. And so I was curious if you could speak a little more to your ideas about what might be driving that and why we're not seeing the effect we might actually predict.
Yeah, that's a great question. So I think it may, so I have two things to say about that, right? So one is, I'd be very curious to know how much it's a developmental difference, right? So it'd be really interesting to do a study where you do kind of the parallel thing across multiple ages in development and seeing if there's a crossover point, right? That's one hypothesis. A second hypothesis is it a kind of a depression severity issue? So, kids who have depression by preschool, we tend to find have a pretty heavy family history of depression. Often, issues going on in the family. This may be a particularly, we haven't been able to look at the genetics to know, we just don't have that data to know, like, do they have a particularly high loading? There's not that many SNPs who've been associated with depression yet, but so because the literature on cortisol and depression is so mixed, right? Like some of it's increased morning awakening, some of it's decrease, and we very clearly saw a great, and I should really say it was Mike's work. So I do wonder if it's a developmental difference. I wonder if we would see something different if we use different stimuli. So like in our face processing study, we saw increased amygdala responses to sad faces. So I wondered if maybe there was something specific to the emotion content, because we didn't see that to the fearful faces. It was really to the sad faces. So maybe losing money wasn't sort of the right stimulus, or losing candy, right, wasn't quite the right stimulus to elicit increased amygdala response, right? Maybe, because I almost think of it as in that study that they just things that normally pleasurable don't matter to them, right? And so doesn't matter if I lose Skittles, doesn't matter if I win Skittles, right? I think that's different than maybe saying like, oh, sadness and negative affect and guilt really elicit a stronger response in me, right? So it's possible we would have seen increased amygdala activity if in those same kids, like we'd shown them emotionally evocative films, or, sad faces, or, images of really sad things, because maybe that would tap more into what their emotion dysregulation was.
Hi, I'm Claire. I am a graduate student in the psychology training program here at UW. And my question follows pretty nicely from your response from the previous question. And I wanted to hear more about how you think about the variability in mechanisms that might be at work in childhood onset depression versus potentially the adult depression. I've just always kind of had a gestalt that maybe kids who display depression really, really early in life may have a different set of mechanisms going on or a more similar set of mechanisms going on to each other than adults? And I'd love to just hear how you think about those things.
Yeah. So we haven't yet seen a lot of evidence that is really different from the things we see in adults, right? I mean, we see hippocampal volume reductions. Like we kind of started this in a somewhat boring way, which is like, let's see what's wrong in adults and look at that in kids, right? And so we're not seeing a lot of evidence that there's like these really clearly very qualitatively different mechanisms going on in kids. And we have been trying pretty systematically, you know, to look at different things. So I have become less convinced that it's really qualitatively different. I mean, at the same time, the things that are in kind of really intriguingly different are, so in prior to puberty, we see pretty similar rates of depression in boys and girls. And if anything, we see slightly higher in boys. Whereas at puberty, so what you see in longitudinal studies is like, it's like 2% of the population, very similar in boys and girls. Puberty hits, rates go up in boys and girls, but they go up much more steeper in girls, right? So there's clearly something different going on in adolescent depression where you now start to get these gender differences, right? Which we don't see in the early onset depression. So Catherine Luking, who's now going to be an assistant professor, she's really interested in understanding puberty hormones and how that may play a role. There also seems to be evidence so in the little kids, we haven't looked much at like social aspects of depression, social rejection. We have some data we're collecting and we've looked at it more in adolescence, but there's some suggestion that kind of the social peer information becomes particularly salient to girls early and maybe a particularly salient risk factor for depression onset in girls versus boys early on.
Early on or
Yeah.
Okay, so
Post puberty, right?
Post-puberty,
Not pre. We haven't really looked at it pre puberty, so I can't say that's true. But post puberty, the data already in the literature does suggest that social rejection and peer feedback is particularly salient for girls as a risk factor for depression. So I think there are some differences, right? But the things we started with aren't as much those, right? Because it feels like there should be differences, but we're just not seeing a lot of them.
and what about the idea of variability? Do you think that there's more or less or similar variability in the drivers of depression in kids versus adults?
I don't know. There's so much variability at every point in development. I mean, every form of psychopathology has so much variability, right? So I can't say that our data, our personal data speaks to more or less variability mechanistically in kids versus adolescents versus adults. And I've done work with all of them. You know, it's easy to point to family circumstances in kids, but we also know that adult depression is very likely to be associated with interpersonal disruption too, right? So I just don't see any evidence yet. I'm not, it's more like we have an absence of evidence, not evidence of absence.
Thank you so much.
Yeah.
Hi, my name is Bethany and I'm a graduate student at Iowa State University. Super fascinating study that you were able to get MRI scans, like starting in childhood longitudinally, and then to be able to have children perform the TSST or a version of it and collect the cortisol. It's like awesome. So my question is about puberty. You touched on it briefly in your presentation. You kind of answered a bit of it just now. My question about puberty was that some people consider puberty to be a period of critical development, and during puberty, it's considered a time where the brain undergoes reorganization and these youth start to feel emotional and behavioral changes and differences. And so I was wondering in your study if you included any measures of pubertal development, like the like Tanner staging or things like that, and if you were able to see any associations between pubertal development and mental health outcomes like depression, and then also maybe like structural and functional differences in the brain.
So yes, so I'll tell you what we've already published, and then Catherine has been working on a bunch of papers looking at our hormone data. So I don't want to like scoop her because she hasn't published them yet. But what we've published one. So we started collecting the Peterson puberty scale, which everybody does, right? Which is when we got parent report and youth report of physical puberty maturation. And then we started collecting hormones kind of wave 2 of imaging. Wave 2 and 3, we didn't do a great, we were kind of doing it, we weren't funded to do it. We were kind of doing it on the side. We did it on the day of scan. We got a testosterone, DHEA and estrogen. And then when we were refunded for scans 4 and 5, we did it like the real way, right? Where we timed it to, periods, and we got all the fancy measures for girls of whether they had hormones and so, hormonal birth control and so on. So the one paper we've published so far that kind of speaks to your thing is we actually looked at the relationship between early poverty and trajectories of testosterone and DHEA over a course of development. And what we saw was that greater poverty was associated with a shallower slope of testosterone increase over the course of development, which was in turn associated with a shallower increase in hippocampal volume, which was in turn associated with greater emotion dysregulation and greater depression. And we ran mediation models, suggesting that there was at least some partial mediation of testosterone and hippocampal volume on the relationship between early poverty and later depression and emotion dysregulation. Didn't see it for DHEA, and I didn't look at estrogen. We were really focused because there was so much evidence about kind of neurosteroids and testosterone and DHEA and neurogenesis. We focused on those, although estrogen, is important too. So that's the one paper we have published. Catherine's now been really intensely looking at early puberty and its role as a risk factor. And she would have a lot to say about that. So she definitely has some data consistent with the idea that it's already out in the literature. It's not like we created that idea that early puberty is associated with increased risk for depression. What she's really, really interested now in is moving from, I can never pronounce this right. So there's gonadarchy, is that how you say it? So that's the later phase of puberty, right? That is associated with what we typically think of pubertal physical maturation, voice deepening, breast development. But there's adrenarchy, which is the really early phase of puberty that isn't typically associated with external differences, but you start to see rises in testosterone and DHEA. And so she's really interested in starting to measure that and looking at that as an even more sensitive early indicator of early puberty and sort of pubertal things. So we did this other project called the Human Connectome Project Development, where we got hair samples and saliva down to age 5. And then Catherine also has another project going now where we're collecting new data. Really, she's collecting new data in these much younger kids to look at adrenarchy and see whether you can start to see before what we typically think of as pubertal onset, kind of whether greater increases as a function of age in pubertal hormones are kind of preceding girls in particular showing increases in emotion dysregulation, which then might precede the onset of depression. That's all. We don't have, she has a little bit of data consistent with that, but we don't have the hormones in the really young kids to test that yet.
Thank you so much.
Hi, my name is Matt Peverell. I'm a postdoc here in the Department of Psychiatry. Thank you for this amazing talk, a really incredible body of work. I had a question a little bit related to Claire's a moment ago, which is just one of the hopes I think many people have had for the study of psychopathology in the brain is that we could try to sort of disentangle like multifinality and like different kinds of brain systems that might represent separate phenotypes leading to depression. You found impacts or correlations between a variety of different brain systems and depressive symptomatology and risk in children. Just wondered, do you have any hypotheses about whether these are all sort of present in higher risk children, or are there sort of individual presentations where one brain system is sort of more disrupted than the others?
That's a really great question. So I think in our preschool depression and PCIT sample, they're tending to go along together. The same kids are having these disruptions, but they're not very big samples to do this kind of multifinality work. And I know there are people trying to tackle this like in the ABCD study. I've been on the dissertation committee of a really bright neuroscience student who's trying to do this in older adults, right? From 2 perspectives, right? So one is there phenotypic so you could ask this question in multiple ways, right? You could say, are there really subtypes of depression with sort of non-overlap, where there's a neural system and a phenotype that goes together, and those are different, right? You could ask the question of whether there's different subtypes of neural patterns associated with the same phenotype, right? And then you could ask whether there's the same neural disruptions associated with different phenotypes, right? All three of those are possible, and they are going to really require much larger sample sizes, because in part what you have to do is, people don't come cleanly with different things, right? So you need large enough samples where you can sort of create artificial subgroups of like, so for example, one of the things she's going to look at is, well, what if you look at people who are high on anhedonia but low on depression versus people who are high on depression but low on anhedonia, right? Are those phenotypes that are associated with sort of fundamentally different neural systems, right? And the problem is, like in our samples, like it's not easy to find enough kids who have that, right? But she's using like the UK Biobank, where there's thousands and thousands of people, and she can kind of pull out artificial subtypes to ask whether there might be these things there. So I think it's a really interesting question, but it's really hard in kids to get big enough samples to do that well.
Thank you.
Hi, my name's Ryan. I'm a postdoc in the psychology department. I also do a lot of EEG work. I was wondering, you have some kind of neurological markers of reward processing across these timelines. And you also, I believe, showed decreases in anhedonia, which has a lot to do with reacting to reward and experiencing reward and things. And I was wondering if you found relationships between those changes in anhedonia as a symptomology and the neural markers of reward processing.
Modest. It was like a correlation of 0.2 in the clinical ratings of anhedonia and the neural markers. So it wasn't super strong, but it was also a single item anhedonia measure. But it was certainly something we wanted to look at. So I would have liked it to be stronger, but I don't think we had great measures of anhedonia in the kids. That's my out. Yeah.
Thanks.
Hi, my name's Mike. I'm a postdoc at McLean Hospital and Harvard Medical School. And I want to echo what others said about just that. I think it's truly excellent how you're able to harness parents to be able to treat kids. I have a background in public health, so I think a lot about how we can reach as many people as possible in light of the shortage of healthcare providers and mental health providers. And I really like the direction that you alluded to of trying to take a more community-based approach in schools. Of course, oftentimes even really excellent school-based programs are underfunded and don't reach nearly as many communities as we need to. So in light of that, my question is, I'd like to hear your thoughts on the potential feasibility of maybe an app or something electronic that would be able to reach parents who then in turn could reach the kids, and related to that, to maybe help all of us understand the challenges. I'm interested in getting some sense of what's required from the parents to be able to really do this, like how many hours it takes for them to learn and all that.
Yeah, that's a really good question. I haven't really thought about an app-based approach. So I'd have to think about it before thinking about whether that would work. I mean, in the full-on PCAT, at minimum, it required them trying to come in for 18-hour-long sessions, plus doing homework. And not everybody did homework, and not everybody came to every session. So that's a non-trivial expectation on the part of the parent. What would actually in some ways be a little, I have no idea how you would do this, right? But this relates a little bit to Katie's question, right? Is, part of it is like, there are times when the parent really, if you're thinking about it more reactively, there are triggers where the parent really should intervene and help the kid regulate, right? And sometimes one of the challenges that parents have is not recognizing, right? And I'm a parent, I had those problems too, that like, now's the time that I should start helping this kid, either upregulate positive emotions or downregulate negative emotions. So I think of like tools, like there's this thing called Lena, where they have babies wear these things and you can capture all this speech. Like it would be really interesting if you had some machine learning algorithm that could detect when kids were starting to become dysregulated and like text the parent and be like, now's the time to go help your kid, right? I have no idea how to make that happen, right? But like that is the kind of thing where I think, boy, that would be really interesting, but I have no idea how to make that happen, right? Or what you'd have to have a lot more data about like what was like life like in the home and when are the parents and kids together or what are the trigger points for the kids. And what I will say is the person who asked about variability, I mean, with kids, there are, of course, standard things that often trigger their emotion dysregulation, but there's also a lot of variability in the specifics per kid. So how would you be able to come up with something that was general enough to capture all those individual differences? And, is it peer relationships? Is it around toys? It is around food, TV, where are the trigger points that get this kid going?
It sounds like maybe a within subjects approach would be.
Yes,
really
you
key
could
to be able to-
imagine some ideographic kind of approach that might be really interesting.
Yeah,
Yeah.
and it's not my expertise yet. I'm starting to move into that direction with machine
Great.
learning.
Okay, you could
Um,
solve that
yeah.
problem.
Yeah, I recently got a small fellowship looking at something similar to that, but with fear learning.
Yeah.
It's a fascinating and very challenging task, but there are some people doing some extremely
Yeah.
impressive
Yep.
work with N of 1 approaches. So
Yep,
I'm
exactly.
hopeful that in time. But thanks so much. That's really
Sure thing.
fascinating and promising.
Okay, great. Do we need to stop or do we have time for more? We got 2 people, so that's perfect.
Well, thanks, Dr. Barch, for this talk. My name is Lana. I wanted to actually continue the discussion with the focus on the parents. And so I had 2 questions. I was curious if in your data, the report of the child's depression symptoms are coming from the child, the parent, or a clinician, and whether the parent report maps on well to a child or a clinical report and the outcomes that you discussed.
Okay, great question. So the answer to that depends a little bit on the study. So prior to like age 7 or 8, we almost exclusively rely on parent and clinician and teacher reports. We'll start to add in kid self-report, usually like 8 and older. I will tell you that kid and parents and teacher do not necessarily correspond well for internalizing symptoms. Parents and teachers can be okay on externalizing. Teachers are not very good at reporting on internalizing symptoms, right? Unless the kid is really showing a lot of negative affect and crying and those sorts of things. So teachers, really good at externalizing symptoms. Parents are good at externalizing symptoms, can report on internalizing. We see when we have, the clinicians, we have clinician ratings, but of course the clinicians are interviewing the parents. So yeah, there is a correspondence because the information that the clinician is using, although the clinicians in our studies are typically also getting information from other sources if they can. They're observing the child, they're talking to the child, they're not asking them about their mood, but they're like, and we do all these like observational tasks and things too. So the clinician has more information than just the parent report. But definitely when the kids get older, then we start asking the kids. And, the data in the literature suggests that age 9 is about when reports of internalizing start to diverge between parent and kid for both like symptoms and like suicidal ideation. The literature is not super strong, but some of it does start to suggest that the kid report is going to become increasingly important for internalizing because they are having access to this whole internal world that they may no longer be sharing with their parent. So.
That sounds so important for the aspect of early intervention too.
Yeah.
If it's the parent who has to get the child into the treatment.
Yeah.
And then just a little more broadly, I was curious as to what you might think the potential impact could be of more societal level interventions to support the parents, like having greater paid leave, daycare.
I think it could be great. And I think you should all join me in lobbying Congress about that. We actually we have written a few op-eds and Joan, is very adamant about this. Like she and one of our postdocs wrote an op-ed when they were considering which thing, extending the child care tax credit. Like, I mean, there's a lot of things I think we could be, and it'd be really interesting to talk about like the Netherlands and other places where we do have much more supports for parents in a lot of these ways, and to do some more comparative studies, because people always say, well, how does that compare to, and often it's, European countries that have better supports for kids. And there haven't been a lot of recent epidemiological studies to say, for example, are there much lower rates of childhood depression in kids or not, right? But Joan would have a lot to say. Yeah, so.
Hi, I'm Andrea Stein. I'm a grad student here. So my question was kind of sparked by that very first study you just talked about, showing different patterns of activation in response to sad faces. And my question is, given so many depressed kids have depressed parents, whether you have any data or intuitions as to kind of what piece of that different reactivity to negative stimuli, in particular negative faces, might be attributable to different environmental exposures from the parents versus.
Yeah, that's a great question. We don't have any data that speaks to that. I wouldn't be at all surprised, right? Because, we see that something like 90% of our moms who brought kids in for early onset depression report having their own past history of depression, right? And there's a whole other literature looking at, like early depression in moms, and how, unfortunately that data seems to suggest that treating depression in moms doesn't necessarily improve the outcomes for the kids early on. But I wouldn't be surprised, but we don't have any data that would speak to that directly, but it's a great question.
Thank you.
Thank you so much, Dan.
Okay.